Hello, everyone, and welcome to Lunch Break Science. I'm Arielle Johnson, your host from the Leakey Foundation, a nonprofit that supports human evolution research and shares discoveries. Today, we explore what genomics tells us about primate digestion and take a look at eating insects. Before we dig deeper, thank you to the Ann and Gordon Getty Foundation, Camilla and George Smith, and the Joan and Arnold Travis Education Fund who made today's episode possible. Now let's bring on our guest, Dr. Mareike Janiak, who is a Leakey Foundation grantee. Mareike, thank you so much for being here with us.
Yeah, thanks for having me.
Mareike is joining us from Montreal and is a postdoctoral research consultant with the Hayam Lab at New York University, the Snyder-Mackler Lab at Arizona State University, and the Moline Lab at the University of Calgary. Her work has taken her to field sites all over the world. We just flew past Costa Rica and are now seeing Cayo Santiago, a very unique island in Puerto Rico. And now we are headed across the ocean to the Simien Mountains of Ethiopia. If you're watching live, tell us where you're joining from in the chat and what questions you have for Mareike. The earlier you get your questions in, the more likely it'll be featured. Mareike, why do you use genomics to learn about primate digestion?
Yeah, it's a great question. So the main reason that we use genomics is that it's a non-invasive method. So a lot of other fields and a lot of research into digestion is a lot more invasive. So they might cut open an animal, they might take samples from the inside of a stomach. And those are things that those of us who study primates, generally, we try to avoid for several reasons. One main reason is that primates are really endangered. So you often, you can't get permits or permission to do these invasive types of analyses or sampling with primates. And, not just that it's illegal and or hard, not just illegal, but hard to do. It's also that most of us try not to do these things. We try to study primates non-invasively. And genomics is just a really good way for understanding dietary adaptations in a way that doesn't require intensive invasive sampling. We can do this from a small tissue sample or even a fecal sample.
Well, your talk today is an excellent look at applying genomics to a question, and I'm really excited to learn if monkeys and humans can digest insects. So let's start and hear about some of your other projects after your presentation.
Yeah, great.
Before Mareike starts, though, if you want to learn more about human evolution, amazing research going on all over the world, and the latest discoveries, start by subscribing to our channel and clicking the notification bell so you don't miss a thing. Now, let's turn the virtual floor over to Mareike and learn about insect digestion, or digesting insects, not insect digestion.
Yeah, so I'm going to just give a brief example of how I can use genomics and genetics to understand more about dietary adaptations in primates. So this was work that I did for my dissertation research. And, I was interested in how different types of primates have adapted to eating different kinds of foods. That's where I was coming from, because primates have just this amazing diversity of foods that they eat. Some primates eat really, really tough seeds. Some really focus on fruit. Others really focus on leaves, which have their own challenges. Some primates eat fungi. Geladas only eat grass, which is a very strange diet for a primate, but also insects. A lot of primates eat insects. And I kind of hinted at this when I talked about the leaves, but all these different foods have different challenges. So leaves, for example, they have cell walls that are made up of cellulose, and cellulose is something that our bodies on their own can't digest. We don't produce an enzyme that can break down that cellulose. So any primate or other mammal that eats cellulose or leaves will digest those with the help of microbes in their gut. Insects, on the other hand, they don't have cellulose, but their cell walls are made up of something called chitin, So they're exoskeletons, which you're familiar with bugs. They have really tough exoskeletons. Those are made up of chitin. And that's also another challenging component that we don't know for sure. We didn't know for sure if primates could digest. So this in the corner is a tarsier. It's eating an insect. But a lot of other primates will eat insects either as a large component of their diet or a smaller component. And this includes some of our closest relatives, chimpanzees. And chimpanzees are really famous for eating insects in a very particular way. And we have a little video to show this. So this is something that Jane Goodall actually observed when she was studying the chimpanzees at Gombe. And this is called termite fishing. So chimpanzees will make a little tool out of a little stick that they then use to dip into the termite mound and fish out insects. And I think in the next video, you can see this very clearly. You can see how the little termites are hanging onto the stick, and the chimpanzees then just going along and kind of picking them off the stick. And like I said, insects are challenging because they're covered in these tough exoskeletons that are made up of chitin. Now, for a chimpanzee or something else that's really large, that's not going to be that big of a challenge because, you might not be able to digest the exoskeleton, but, we have a large gut, you have room for that exoskeleton to hang out and just get past along with any other indigestible things. So for chimpanzees, it's not a big deal, but for something like a tarsier, and this is a very, very small bodied primate that eats almost exclusively insects. You can kind of see the relationship between the size of the insect here and the size of the tarsier it's trying to eat the insect. So imagine you're a tarsier that is eating these insects and you can't digest the exoskeleton. That is just indigestible bulk for you. And your only mechanism for dealing with this might be to just pass the exoskeleton quickly and poop it out, and then get the nutrition from the rest of the insect. But that's what for a really long time was kind of the prevailing idea in primatology and in just general mammalian science, was that mammals couldn't process these insect exoskeletons, and they were dealing with them mostly by just passing them quickly. And to me, that was kind of counterintuitive because, if you're such a small-bodied primate like a tarsier, can you really afford to have a bunch of indigestible bulk in your gut and not get energy from this large chunk of the insect you're eating? So I kind of, came to wonder, maybe that's not actually true. Maybe they have mechanisms to digest these exoskeletons. And the mechanism that I focused on, that I was curious about, was digestive enzymes. So these are enzymes that are produced in your stomach, in your pancreas, different parts of your digestive tract that help you break down foods. And some really well-known ones are lactase, sucrase, and amylase. So these are different types of enzymes that are specialized for breaking down sugars in the food you eat into smaller components. So for example, the main sugar in milk is lactose, which is a large sugar, and your body can't absorb that on its own in the form that it is in the milk. It first has to get broken down into smaller components. So in the case of lactose, the lactase enzyme basically cuts that big sugar into two smaller sugars, glucose and galactose, and your body then absorbs those. And the same is true for different types of other foods. So I was curious, maybe there is an enzyme that's produced in primate stomachs, or in the pancreas or somewhere else in the digestive system that breaks down chitin. So, this might be a chitinase. And I was really lucky that around the time when I was getting interested in this, some research came out on insectivorous bats. So in this study, the researchers took actual samples from along the digestive tract of these insectivorous bats, and they put them in a petri dish. And this is the petri dish here. And you can see there's a silvery substrate or bottom of the petri dish. And that is the chitin. So this is just a lab type of chitin, which is structurally very much like the chitin you would find in an insect exoskeleton. And then they dropped samples from different parts of the bat stomachs in these 6 locations you see labeled on this Petri dish. And you can kind of see that there's areas around where samples have been dropped that have turned a darker gray color. So the first one is a reference chitinase, a standard chitinase. So this is, again, a lab quality chitinase. So this is not from an animal. This is just a reference. So this is to see what it would look like if there was chitin digesting activity in the sample. So you can see there's a darker gray circle around where the sample was dropped. And you can see the same thing is happening next to the sample from the stomach of the bats. So there's a darker gray circles going around the stomach sample. So this shows that there is chitin digesting activity going on in the stomach of insectivorous bats. This was also done in mice, which I'm not showing results from that here, but similar results have been found for mice. So this is really, really interesting for me because, here I have an example of another mammal that's eating insects and really can't afford to have tons of exoskeletons that it can't digest in its stomach because it has to fly, and you can't really be weighed down with all of that when you're trying to fly. So that gave me an angle to look at in primates. And the really useful thing for me was that I was able to find out what gene codes for this chitinase. And that was important for my primate research because, I couldn't do what these bat researchers had done and sample the actual stomach contents from different primates. So I was trying to get at this purely from a genomic perspective. And so the gene that codes for this chitinase is called chia. If you are familiar with chia pets, it's easy to remember that way. And so my goal was to kind of see if primates had this gene, if it was a functional gene, and look at this in a variety of different primates that eat different types of foods. And my hypothesis was basically that most primates might have one of these chitinases, might have one of these functional chitinase genes, because you will then produce the enzyme. And most primates have some insects in their diet. But as you maybe increase the amount of insects in the diet of these primates, maybe you'd see that there were multiple genes for this, because then you would produce more of this enzyme. Whereas if you're a primate that doesn't eat any insects, some of the monkeys only eat leaves, for example, don't have insects in their diet, then you might see that the gene is no longer functional because there's no need for this enzyme, so you wouldn't produce something that you're not going to use because that's not efficient. So with insects, this is kind of the idea. The more insects you eat, you might have more of these genes, whereas if you don't have any insects in your diet, you might not have a functional gene for this enzyme anymore. So I did this through a combination of sequencing. So I actually sequenced this gene in a number of primates, which looks something like this. And then I also mined genomes of primates. So these were assembled genomes that are publicly available. And I was able to search for this gene in these genomes through bioinformatic analyses. And that looks less glamorous. You basically just stare at a screen called the terminal, which is where you do this work on the command line. And so to my results, I was pleased to see that most primates did have this chitinase gene, and it appeared to work. There were no signs or mutations that indicated that it wouldn't. So most primates followed what I predicted, that they eat some insects and they have one of these chitinase genes. And I also found that, like I predicted, that some primates didn't have a functional gene for this anymore. I could find the gene, but there were mutations in it that suggest that it's not functioning anymore. And this was found mostly in primates that don't eat insects. So these were frulivorous primates, or the grass-eating primate, the gelada, that didn't have a functional version of this gene. But I also found that some primates had multiple copies of this gene, and they were primates that ate more insects. So these were mostly monkeys from the Americas, also called platyrrhines. And here I found 2 copies of this chitinase gene, and they were also more insectivorous. But it didn't end there. I actually found that some of the species in my sample had 3 of these genes. One of them is a primate, a bush baby, which also is very insectivorous. And the other one was a tree shrew, which is not a primate, but a close relative that also eats insects. And the big surprise that I found, which fit with my hypotheses, but was a very, very nice result to see, was that the tarsier, the most insectivorous primate, had 5 different chitinase genes. So it really follows this hypothesis that the more insects you have in your diet, the more functional genes for this chitinase enzyme you might have. So overall, the results suggest that, like insectivorous bats and mice, primates might produce an enzyme that helps them break down the exoskeletons of the insects that they eat, which would be a really important adaptation because it will allow them to digest all of this insect material properly and get energy from it, as opposed to just passing it really quickly. So the question that I always get once I present this is people want to know, well, what about me? What about humans? Do we have a chitinase gene, and can we digest insects? And the answer is yes, we do have a chitinase gene. There's some debate over whether or not we express it in the stomach and whether or not it's actually functional, but we do have the gene. And we also know that insects were a really important food source for some of the human ancestors or cousins. So for example, Paranthropus boisei there's been evidence found that they used tools to dig into termite mounds, not exactly like chimpanzees, a little bit more in a destructive manner, but somewhat similar, that they might have relied on termites as a food source. And let's not forget that even though in the Americas or North America, like Canada, where I am in the US, we don't eat insects on a regular basis. They're not really part of our diet. But around the world, many different populations eat insects pretty regularly. They're a standard part of the diet. So it is not weird for humans to be eating insects. It just seems weird to us maybe here. But generally, it's something that we very well can digest. And there's a case to be made that insects are a food that should be embracing more here in the US and Canada, because beef and general livestock is very destructive for the planet. A lot of deforestation is due to cattle ranching, for example. The overall biomass of mammals on Earth is dominated by livestock. And also livestock is using a disproportionate amount of water for producing foods. So insects might be a really good alternative if you want to cut down your meat consumption, but you're worried about your nutritional intake. Insects might be a great alternative here. So they have tons of protein, lots of vitamins, all 9 essential amino acids, and they use a fraction of the land mass and water to produce. So if you feel like embracing your inner tamarin, I suggest that you give insects a shot. And I want to thank everyone who's involved with this work and all the funders that made it possible.
Thank you so much, Mareike. That was really fascinating. Tarsiers are just incredible, and I know you would love to work with them again. What is your dream project?
Yeah, so the thing about the genomic research is that in a lot of ways we're generating hypotheses, because we have found these 5 different chitinase genes in the tarsier, but from these data we don't know where in the body they're expressed necessarily. We can infer that they might be expressed in the stomach, but we don't know that for sure. And also all the 5 different chitinase genes in the tarsier had subtle differences. So I'm really curious, do these differences matter when it comes to digesting insects? Do they all digest them equally efficiently? Are they, targeted at different types of insects? Who knows? Maybe they're, working in different parts of the body. Maybe they're in the saliva to kind of get a jump start in the digestion. So, you know, there's all kinds of different things that could be going on here. And I would love to, you know, either maybe clone these different enzymes into a bacterial vector, which is something you could do, where you then kind of in a petri dish can test how well they're digesting insects. So yeah, there's different ideas I have for how I could look at this in the future. So that would be kind of a dream project.
Awesome. We have a couple of questions. One is from Lori. Has the chia gene been discovered in the Neanderthal or Denisovan genomes?
That is an excellent question. I don't know the answer to that because I haven't looked at that, but you've just given me a wonderful idea for something to do. Yeah, and you know, also like if anyone is at all technically minded, these genomes are publicly available. So there are tools you can use on the web to do this yourself. It's not always super easy, but yeah, if anyone is kind of technically minded and curious, I would encourage you to look up these online resources. So Ensemble, for example, is a genome browser where you can kind of look at genomes of Neanderthals.
Well, we'll have to get some of those links in our video description.
Yeah.
But thank you for sharing. I'm like now I know what I'll do this weekend. I have another question from Pete. Hi Pete. Thank you for your question. He says, right now I'm thinking nocturnal primates are more likely to eat insects. Is that so?
Yeah, I think that's generally something I would say is true. Tarsiers, for one, great example of a nocturnal primate that eats insects. In bush babies, another example, so it would be an African primate. In the Americas, we only have one primate that is nocturnal, and that is the owl monkey. And they do eat some insects. Of course, with nocturnal primates, it's often really difficult to know for sure what they eat because they're so difficult to study. But yeah, we definitely think that owl monkeys eat some insects at least. And yeah, I think generally, I would say you could say that nocturnal primates tend to be more insectivorous.
So you didn't start out working on the genome, though.
Nope.
How did you get your start?
Yeah, that's a good point. I started out really doing a lot more field work. So, you know, I became interested in primates and in anthropology first when I was an undergrad at the University of Texas in Austin, because I was really a psych major. It's very different. And I was interested in animal behavior, but I didn't know anything about anthropology and I didn't really know anything about primates. And I just happened to take an anthropology class because I needed another science credit. So I took an intro to human evolution class or intro to biological anthropology. I can't remember what the exact title was with Dr. Chris Kirk. And he was just such a wonderful professor and such a wonderful teacher. And he was just infectious about his enthusiasm for anthropology. So yeah, I got really interested in primates and evolution and primate behavior. And that's kind of how I started out. And then I did a field school when I was finishing my undergrad in Costa Rica. And that's how I went to the field for the first time. This was with Amy Schreier, who was then my instructor and has since become a good friend. And then I kind of went into grad school, but I continued doing some field work in Costa Rica and Kenya also. But then I also got a chance to go to Borneo in Indonesia at Dr. Erin Vogel's field site in Tuanan, where I was working. I worked for her as a TA for her field school that she ran one summer. But I was very lucky to travel to a lot of different places. And then during grad school is when I became interested in dietary adaptations, largely thanks to Dr. Erin Vogel. But then, kind of got into genomics as a tool for studying these things non-invasively. So during grad school, I then learned a lot of lab work, did a lot of these extractions and some sequencing, and also learned a lot more about how to analyze these data.
Oh, it's so fun to see the pictures. So now let's actually take a look at a couple of your other projects. First, let's go to the Simien Mountains in Ethiopia and hear about your work with the gelato monkeys.
Yeah. So I work with a project that is led by Dr. Snyder-Mackler at ASU, with Kenny Chu, who's a postdoc there, and Elise Banyel, who's also a postdoc there. And I sadly have never been to the Sibia Mountains, but I would love to go one day. But yeah, together with Kenny and Elise and Noah, we have published a paper on the high altitude adaptations of the gelada. Because gelada monkeys are unique primates in many ways because they eat grass, which is a really strange diet for a primate to have. But they also live at really, really high altitudes. So you can kind of see that there's mountains here. Excuse me. But they really are on these plateaus, high altitude plateaus. And you can kind of see the cliff dropping off towards the end of the video there. Yeah, so we've looked at high altitude adaptations and then also their microbiome, because to digest all this grass, they must have really unique microbes living in their gut that help them digest these grasses. And Elise specifically is interested in how these microbes might be transmitted from the mother to the infant.
So you've also worked in Costa Rica. What is it like to work there?
Yeah, so I really love Costa Rica. It's probably my favorite place to travel. It's where I went first for my fieldwork, but then we have an ongoing project there with this is with Dr. Amanda Malin and Dr. Carrie Bayeux, who we also have a picture of, who I started working with during my postdoc. So I did a postdoc at the University of Calgary with Dr. Amanda Malin, and then together with her and Dr. Carrie Bayeux, we have established a collaboration with Kids Saving the Rainforest, which is a wildlife sanctuary in Costa Rica. So we often have animals in Costa Rica that might be hit by cars or get electrocuted by power lines. And, if they're found, they're brought to this wildlife sanctuary to hopefully be rehabilitated. So they have vets working there that try to heal the animals and then release them back into the wild. If that can't be done, then they usually live at the sanctuary. And then in some cases, unfortunately, you can't save the animal, and the most humane thing to do is to euthanize. So if that's what happens, the vets collect samples for us. So we don't harm the animals for our research, but we try to just make the best out of a really sad situation, and to get samples from these animals that would otherwise just go to waste. So yeah, that's what we've established, and this is part of our Leakey Foundation grant too, that we're interested in how different animals adapt for different diets. So especially in these adaptive radiations that we see with the platyrians, monkeys of the Americas. How are these different primates that eat different types of diets adapting? Like, are their digestive enzymes different, for example? And Carrie is really interested in touch. So like if you're eating fruit, for example, like, do you have different receptors in your fingers? Because you might be palpating the fruit to see if it's ripe, or your olfaction, like, do you smell things differently when you're relying on fruit? Because you have to kind of figure out, is this fruit ripe by sniffing it? Whereas if you're eating leaves, that might be less important.
And last but not least, certainly, you also work at Cayo Santiago, which is a very unique site.
So Cayo Santiago is a really wonderful place. So this is work that I have been doing as a consultant with Dr. James Heim at New York University. And the site is off the coast of Puerto Rico. It is run by the Caribbean Primate Research Center. And it's inhabited by rhesus macaques, which are definitely not a monkey that is native to Puerto Rico. They're actually from Asia, and these macaques were brought from India to Puerto Rico in the 1930s. So the colony was established there as a breeding colony in the 1930s, and they've been living there ever since. Obviously not the exact same monkeys, but they're descendants. So the population has grown to roughly 1,700, and they live in these naturalistic social groups on the island and are almost like a natural experiment. So the really amazing thing about Cayo Santiago, in my opinion, is that it has brought together researchers working on a really wide variety of different topics. So what has happened is that all of these researchers have been working together and collecting different types of samples to build up just a database of really wonderful samples. Like we have lots of different types of samples for microbiome work, for genomics work, for any other kind of more lab work like that. But we have paired that also with behavioral data. We have cognitive data. So you have neuroscientists working there. You have just such a wide variety of people working together that really changes the types of questions that you can answer in a primate, whereas in most field sites, you just don't have that. So to me, that's what makes Caio Santiago really special.
Well, we've talked a lot about data, but what is the size of the data you are looking at, and how do you read it?
Yeah, that's a really good question. So with genomics, a big part of the struggle is that you're dealing with sizes of data that are just not what a normal computer can handle. So for example, if we want to sequence a human genome or any primate genome, they're roughly similar sizes. A genome has roughly 2.5 to 3 billion bases, so that's letters. And we want to not just sequence each of these letters once, because then if there's a mistake, you can't be really sure, is it the right base or is it just a mistake? So we want to read every single letter roughly 30 times. So that's kind of like the gold standard is that each letter in the genome is read 30 different times. So if you look at 3 billion different letters and you want to read each one of them 30 times, the numbers get really big really fast. And these sequences that we usually have are kind of like around 150 letters long. So when we want to read each part of the genome 30 times, we quickly get to large numbers like 300 or 600 million sequences. And that can then take up a lot of space, especially when you're not just interested in a single genome, but you want to compare genomes from a bunch of different primates. So for my postdoctoral research at the University of Salford, we had a project where we had sequenced the genomes of roughly 200 primates. These are different, sometimes same species, but different individuals. So we had 200 genomes total that we had sequenced to this 30 times coverage that I was just talking about. And as a result, we ended up with 12 terabytes of data. And this data is already compressed. So the compressed size was 12 terabytes. And just to kind of give you an idea of what that means, what 12 terabytes of data looks like, it's not something you could store on a regular laptop. So the laptop I'm using currently is a MacBook Pro that has 250 gigabytes of data. And to give you an idea, 12 terabytes, I would need 48 of these MacBook Pro laptops to store these 12 terabytes of data that we had for this primate project at the University of Salford. So of course, that's not practical. So all of our work is actually done on what we call high performance computing clusters. So these are kind of like really big collections of supercomputers that are housed often at a university that we access remotely. So, for example, I would use my computer not to do the analyses on my computer itself, but it's basically just a portal through which I access the supercomputer that could be anywhere in the world. So that, it might be in the UK, it might be in Arizona, it might be in New York, it doesn't matter. I can access it from a computer here and do all my work remotely, basically on these supercomputers. And then I think we have an example of what a sequencing file looks like. Yeah, so what you see here is called a FASTQ file. So this is kind of a very typical type of file that you would get from the sequencing machine after a little bit of processing. And in this file, you can have hundreds of thousands up to millions of sequences. And the way they're organized, each sequence is basically given across 4 different lines. So this is just an example of one of these 4 lines. And imagine that these will just repeat over and over again for each different sequence. And they always start with a header line. So that just gives you some information about the sequencing machine and the sample you're looking at. So it's just information. Then the second line is the actual sequence. So these are the A's, C, T's, and G's that make up your DNA. So this is an actual chunk of the DNA that was read. Then you have a little break line. And then the fourth line is a quality score. So this tells you what the quality of each of these letters is. Can you trust that read? Like, can you trust that this is the correct letter? So that quality score, which I realize here it just looks like a random assortment of letters and slashes, but that's an encoding, and the computer knows what quality each of these stands for. So yeah, we can then use these quality scores to make decisions about how to do quality control on our data. So if you see that the quality scores aren't so great, you might do something about that. So here is what that looks like if you visualize it. So this is across all the different reads in a file. And you can see it started out pretty high, all in the green, and then it drops down a bit into more of the orange area. And then towards the very end of the sequence, we'll see that it might drop down almost into the red, and that's what we don't want. So in this case, we might decide that this read towards the end isn't so high quality, so we might remove or trim that part and just keep the parts of the data that we can trust fully. So these are all things that kind of you start out with a new sequencing project, a lot of quality control.
That is so awesome. And actually, I want to quickly give a shout out. We have, for this episode, have raised $220 for our Lunch Break Science fundraiser. So I just want to thank all of our kind supporters. We have a banner to thank them. Yeah, there we go. Thank you, Meredith, Bone Studios, Cheryl, and H. So thank you so much. And now, going back to the data, what is an example of how you analyze this data?
Yeah, so there's so many different things you can do with genomic data. We can use it for microbiome studies where you're looking at what kind of microbes are in your gut, for example. You can use it for genomic studies like we did with the gelato monkeys, where we look at adaptations for high altitude. So then you would look at specific genes. But another example is more like an environmental DNA approach. So when an animal goes to eat something or drink water somewhere, it's always going to leave behind traces of its DNA. And with Amanda Moline at the University of Calgary, she had a little project where we were interested in studying what visits the traveler palm. And the traveler's palm is a palm in the genus Ravenella that produces a nectar that's potentially alcoholic. So it smells a little bit like ethanol. And we think that it might be a fermented nectar. But we know that different types of animals go to visit that nectar and feed on the nectar. So for example, eyes are an example of a primate that visits this nectar and feeds on this nectar. Yeah, so this is an eye. They're nocturnal primates. I think they're super cool. But yeah, we know that they feed on this nectar, which is, potentially alcoholic and fermented. So what we had was samples from this nectar that we sequenced just to see all the DNA that's in this. And something that you can then do is take these sequences and you map them against a database. And this is to try to figure out whose DNA is in the sample. And there could be microbial DNA, that could be fungal DNA, it could be all kinds of different DNA, but it can also be DNA from animals. And, we're hoping to kind of see, well, what visits this plant? What feeds on this besides the ii? Or can we even find evidence of the ii visiting this plant? And this is also a bit of a cautionary tale for having a scientist behind the data, not just letting a computer do these analyses for you and spit out results. Because something that we found when we mapped against these databases, the result that we got back was a little bit unexpected. So when we had kind of our result and our little schematic of what was in our data, something that showed up was entirely unexpected and definitely not what we thought we'd see in a plant that is on land in Madagascar, and that was an octopus. And not just any octopus, but an octopus that's found off the coast of California, which is highly unlikely. We don't think that this octopus was actually visiting this plant in Madagascar. But more likely what's happening is that this is the closest match that was in the database to the animal that we actually sequenced. So a more likely explanation might be that it's a different type of mollusk, like a snail, for example. So this is a snail that is found in Madagascar on land. So that is more likely explanation of what might have been visiting this plant. But yeah, if you just took the data kind of at face value and were like, oh, there's an octopus, yes, an octopus visited this plant, that's just, you have to kind of think about what you're seeing and really like critically and be a little bit skeptical and like think about what your results are really telling you.
Well, that was a really fascinating example of how data can,
lead you astray.
Yeah, run amok. So now let's take some questions from our viewers, because I know we have a bunch out there. If you have not submitted your question yet, please be sure to submit your question now, and we will take our first question, which comes from let's see here, Jeannie. Are there any ratios figured between the percentage of insects as a protein source and body weight?
It's an interesting question. So I think if I'm understanding the question correctly, is there a ratio of how much of your protein can come from insects based on your body weight? So there is a very well-known ratio in primate studies it's kind of a ratio of how big can you be and still be an insectivore. So it's this idea that as you become larger bodied, it's really impossible for you to meet your nutritional demands through insects because insects are small, they're sparse, you have to hunt for them. And, when you get to body size, that's on the traditional threshold that, was proposed as 500 grams. But let's say 500 grams to about a kilogram, you're too big to really feed on insects as an exclusive food source because you wouldn't have enough time in the day to find enough insects to meet your nutritional demands. So just on an absolute basis, it would be impossible for you to do that. So, and we do see in primatology that generally the primates that rely most heavily on insects as a food source are the primates that are below 500 grams to roughly a kilogram. So the smaller you are as a primate, the more likely is it that you would be eating insects. There are some exceptions to this. So one exception that we showed where, chimpanzees during some parts of the year will rely very heavily on termites, but that is because termites are social insects. They're kind of occurring as a clumped resource that is easy to exploit. Another example of something that will feed on insects despite being a little bit too large technically is a patas monkey. So these are monkeys found in East Africa. And they exploit acacia ants, and acacia ants are found in kind of these bulbous agglomerations on acacia trees. And they're, again, kind of like a clumped snack of a bunch of insects you can kind of pick off and eat. So there are exceptions to this rule, but yeah, generally what we see is that primates that rely on insects as a main food source are below 500 grams to below 1 kilo to 500 grams, yeah.
Our next question comes from Pete. Pete asks, I believe geladas seek human food, or is that another baboon? Although I think it's technically a monkey. Is that affecting their digestion genomics?
Yeah, it's an excellent question. So you might be thinking of baboons, olive baboons specifically. So geladas, they're sometimes people refer to them as gelada baboons, but that's not technically correct. They're gelada monkeys. They are closely related to baboons, but they're not technically considered a baboon. I'm not sure that geladas seek out human food. I haven't heard that, but I could be wrong. I do know that olive baboons and other baboons in in Africa will seek out human food, definitely, and like macaques do the same thing. So whether or not that affects their digestion genomics, it's very clear that it affects their microbiome. So their microbiome does look different. As for their genomes, I think it's a little bit like it hasn't been going on for long enough to really have affected their genes, because there we're really talking about an evolutionary time scale of the organism. So, you know, human, like this has really become more of a problem as we have like large garbage dumps, for example. So, you know, when we have large garbage dumps, that will often attract baboons and they will then just feed on human garbage. So this is a bit more of a recent phenomenon. So we don't really see that yet in their genomes, but in their microbiomes, absolutely, it does change those.
Thank you so much for your question, Pete. Our next question comes from Meredith. And Meredith asks, do you include eyes in your study on insect digestion? They eat a lot of insects, right?
Yeah. So at the time that I did my research on this, I didn't have an II sample and there wasn't a public II genome. So I hadn't included them in that. But I know that you know, there's some new work coming out. I don't want to spoil it, but yeah. So a recent genomic sequencing of IIs shows that they do have chitinase gene, and there's also some really exciting research that a master's student did with one of my collaborators at Kent State University. So in her master's thesis, she had actually gotten stomach samples from the Duke Lemur Center and looked at the RNA. So RNA is a little bit different from DNA in that you're looking at an actual gene product that is expressed. So she was looking to see if there's evidence of an expressed chitinase in the stomach of the II. And like this, I don't think this has been published besides in her master's thesis, but yeah, so she found some evidence that there is a functional chitinase in the II stomach.
Ooh, I'm like, I want to learn more about that.
Yeah, she presented this at an AABA, I believe, maybe in Cleveland. I have a memory of her poster because I thought it was really cool.
Oh, so, and thank you, Meredith, so much for that question and for your donation. My last question for you, Mareike, is do you have any advice for those just starting out or hoping to pursue a career in science?
Oh, that's an excellent question. Yeah, I mean, if you know you're interested in going to grad school, I would highly recommend trying to find some opportunities to work in people's labs. So often when you're an undergrad at a larger university, there will be projects that you can work on in a lab so you can get experience. You might want to look at, if you're interested in field work, you might want to consider going to the field as a research assistant on a project to just get some experience and see what it's really like. Because it's not for everyone, definitely. But yeah, and you know, there's different ways to be a scientist too. Like you don't have to go to grad school, I think, to be a scientist. There's other opportunities to do scientific work that's not necessarily like in the traditional sense of going to grad school.
Well, thank you so much for joining us, Mareike. This was a absolutely wonderful episode.
Yeah, it was a pleasure. Thanks so much for inviting me.
Yeah. Hopefully we'll have you back when you have some exciting publications in the future.
Yeah.
So next time on Lunch Break Science, we celebrate the 50th episode of Lunch Break Science. We will bringing back some familiar faces to discuss misconceptions about human evolution. So we just are so excited and look forward to celebrating with you. So hopefully you'll join us. Thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Until next time, stay hungry for knowledge. Lunch Break Science is brought to you by the Leakey Foundation and made possible by the generous support of the Ann and Gordon Getty Foundation, Camilla and George Smith, and the Joan and Arnold Travis Education Fund. Subscribe to the Leakey Foundation's YouTube channel. Hit the little bell to get notifications. If you like this episode, please be sure to like it and support Lunch Break Science through giving fundraiser. Right now, all donations will be matched by a generous sponsor. Also, subscribe to the Leakey Foundation on YouTube, Twitter, or sign up for our newsletter to be the first to hear about upcoming episodes and programs. Still hungry for science and can't wait till our next episode? Check out Origin Stories, the Leakey Foundation's award-winning podcast, available wherever you listen to podcasts. Visit us at leakeyfoundation.org to learn more about the Leakey Foundation, today's guest scientists, our educational programs, research and grant and scholarship opportunities, human evolution news, and how you can help support human evolution research programs like Lunch Break Science, and much, much more. Thank you for watching, and see you next time.